Well, hello, and welcome to this session on exploring developments in the Alzheimer's therapeutic pipeline. It's been a very interesting past couple of years in Alzheimer's disease, particularly in the therapeutic area. It's been a bit of a mixed bag. We've had some remarkable progress in the development of informative biomarkers that would, using only blood-based technology, would enable the identification of patients with specific kinds of pathology. And this is a really remarkable feat in the neuroscience area. The therapeutic development has also achieved some milestones, but not quite the robust success that would indicate that we are clearly on the path to developing very effective treatments. We've got some things that are very promising, and we know that aducanumab is still under review by the FDA and may have a path toward approval, but the data are not so overwhelming that we're entirely confident that that's going to be the major approach to the treatment of Alzheimer's disease. So with that as a background, it's very important for none of us to diminish our efforts, to slow down our search for new approaches, both on a scientific level, new approaches to clinical development, and new approaches to financing this effort that is so important for advancing the quality of life of patients with Alzheimer's disease. We have a tremendously interesting group of panelists today to help us discuss these issues that face the field broadly, scientifically, financially, and in terms of clinical trials. It should be a very robust discussion. We've asked each presenter to use only one slide so that we can have plenty of time for discussion. And believe me, I've seen the slides and they are very good and they are very helpful for framing this discussion. We'll now ask each one of the panelists first to identify themselves, and our first panelist will be Jeff Cummings. So Jeff, could you give us a bit of your background?
Hi, Richard. Thank you for having me on the panel. It's a pleasure and honor, and thanks to Lausanne for sponsoring this. Fantastic. I'm Jeff Cummings. I'm the director of the Center for Transformative Neuroscience at the University of Nevada, Las Vegas, and the founding director of the Cleveland Clinic Ruvo Center for Brain Health.
Thank you, Jeff. Our next panelist will be Dr. Meiyu Geng of Green Valley Pharmaceuticals.
Hello. Hello, everyone. This is Meiyu Geng. I'm actually the academic deputy of Shanghai Institute of Material Medica, Chinese Academy of Science, and also the principal investigators on the neuropharmacology, and also as a chief scientist in Shanghai Green Valley Pharmaceutical Company. I'm the key inventor of the Genentech, a marine-derived oligosaccharide anti-AD drug, which has already launched in China last December. Thank you.
Thank you, Dr. Geng. Our next presenter will be Gene Kinney.
Hi, Richard. Thank you. It's a pleasure being here today. So my name is Gene Kinney. I'm the CEO of Prothena. Prothena is a biotechnology company that focuses on targeting dysregulated proteins in the context of both neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease, as well as peripheral amyloid diseases. It's a pleasure to be here today. I'm looking forward to the discussion.
Thank you, Dr. Kinney. And our final panelist will be Rachelle Doody.
Hi, everyone. It's really a pleasure to be here with such longstanding friends and colleagues for this important topic. I am the global head of neurodegeneration for Roche and Genentech in late-stage neuroscience. But I did spend a great many years as a clinician and researcher in academic medicine and worked on policy as well as patient care. So this is a topic that will touch upon all of these different aspects and challenges in this disease.
Thank you to all the panelists. So now we'll turn it over to Jeff, and Jeff will give us some background on the current state of the AD pipeline. Jeff?
Thank you, Richard. So if I could have the first slide. This is the pipeline as we presented it earlier this year in our annual review. Just to orient you to the slide, the inner circle are the phase 3 compounds, the middle circle are the phase 2 compounds, and the outer circle are phase 1 compounds as listed in clinicaltrials.gov. In the upper slice of the pie in green are all of the biologics, mostly monoclonal antibodies, but not exclusively. In the slice of the pie in purple on the right are all the disease-modifying small molecules, and on the orange slice of the pie on the left are the symptom-reducing small molecules that includes both cognitive-enhancing agents and the agents that are directed at neuropsychiatric symptoms. The colors of each of the symbols we adopted this year from the CADRO, the Common Alzheimer's Disease Research Ontology, developed by the National Institutes of Health, and identifying the principal processes and pathways that comprise targets for the development of new therapeutics for Alzheimer's disease. The shape of each of the icons tells you the population that's included in the trial. For example, the upward-facing triangle are healthy volunteers that would mostly be found in phase 1 studies, or the square are mild to moderate Alzheimer's disease dementia patients that would be found in phase 2 and phase 3 studies. So there's a lot of information on this figure. There are 121 drugs in clinical trials altogether. That might compare with 3,000 drugs for cancer, for example. So it's a relatively small pipeline, given the fact that it is a huge clinical problem. There are 29 agents in phase 3. Again, not very many, given the high failure rate of compounds in the Alzheimer's disease pipeline. There are 65 in phase 2. So that's the most robust part of the pipeline. There are 27 in phase 1. And I should say that clinicaltrials.gov underrepresents the phase 1 compounds because many phase 1 compounds are tested ex-US. So only about 50% of phase 1 compounds are actually captured by our review of clinicaltrials.gov. 80% of the drugs in clinical trials are for disease-modifying agents. So that's clearly the great emphasis of current therapies. 10% are cognitive-enhancing agents, 10% are neuropsychiatric agents, but 80% are aimed at trying to slow this disease process or delay its onset. So that's a very important set of targets that we're thinking about. In total, 31,000 patients are needed for the currently active clinical trials. I've just identified this as one of the major problems of Alzheimer's disease clinical trials, and that is recruitment. Right now, it takes about twice as long to recruit a trial as it does for the exposure period itself. And that's ridiculous. We need faster trials if we're going to have more drugs and get them to patients sooner. So I think that's one of the major problems that is identified by looking at the pipeline. You do see that there's increasing diversity of the pipeline. Only about 16% of agents, for example, are actually amyloid oriented at this point, and there's been a lot of thought about whether amyloid is the best target. I think it's still a very important target. I also think diversification is important, and that diversification is clearly occurring. Another related issue to recruitment is just the site productivity. When we calculated the site productivity for phase 3 disease-modifying therapies, we found that the average site recruits 0.2 patients per site per month per trial. So the system is simply not large enough to accommodate the number of drugs that need to be tested and the number of trials that need to be conducted. So Richard asked us to phrase a call to action based on the information that we're presenting. So Richard, my call to action is we need a national program to educate and organize and stimulate participation in clinical trials. And I think one possibility is to take this moment of COVID, where we're all hearing about clinical trials, we're all hearing about vaccinations, we're all hearing about the need for diversity in clinical trials. Take that as the platform and say, yes, we need it for COVID, but we need it for all neurological diseases, and use this as a teaching moment in the history of the planet to get people involved in Alzheimer's disease and other types of neurodegenerative disease research. So I'll stop there and be glad to take some questions or hear some comments or have a discussion about this part of the presentation.
Well, thanks very much, Jeff. I couldn't agree more about your points regarding the need to have better infrastructure to speed the path of some of these promising compounds through clinical trials. It's kind of shocking when you see in the era of COVID that these vaccine trials for COVID, 40,000 patients are enrolled in a matter of 2 months. And it's not just one trial, it's 4 or 5 of them enrolling simultaneously. So clearly it can be done if there's a will. Just one question from me. Where do you see most of these drugs that are now in development coming from? Does your analysis allow you to give some idea about whether they're coming mostly from large pharma, from small biotech companies, out of academic labs, or where are these compounds coming from?
Yeah, thanks, Richard. Good question. I don't have the percentages at my fingertips, but I will tell you that most of about half of the later stage trials are major pharma, and about half biotech, with academics, as you would expect, contributing more to the early phase trials, particularly phase 2, and particularly the repurposed agents. We've looked specifically at repurposing. And of course, because they're cheaper and they're generic, that tends to be dominated by the academic investigators, and there are very few generics coming in over on the major pharma side. And this is pretty much what you would expect. So the novel compounds are coming out of biotech and pharma, and the repurposed compounds are all coming out of, or mostly coming out of academics.
I could ask if any of the other panelists have any questions for Jeff.
Yeah, I'd like to raise an issue and see what you think, Jeff. We've both been developing drugs for Alzheimer's for a long time. And year after year, we hear everyone's getting out of this field. We're not going to have any more investment. People are going to leave and give up. My first part of the question is, do you see that? Do you see, you've been doing this particular exercise of mapping the molecules in the field for a long time. Do you see activity waning? And the second point is really about siloing, okay? And when we say a certain percentage of drugs are coming from academic labs, a certain percentage are coming from biotech, from large pharma, if you look at the history of any molecule, often it's been one of each. It's been in several different places and several different locations. Do you see a breakdown in that siloing between where molecules are coming from and who gets involved?
Yeah, great questions, Rachelle. Thanks. So we don't really see a systematic waning of interest or investment or activity in Alzheimer's disease. There are a fewer drugs in the phase across all of the phases this year than there were last year. But we did our 5-year review this year. We've been doing this review for 5 years. And pretty much if you look across the 5 years, it's pretty stable. So there was an exit of our pharmaceutical companies from CNS in general, right? And this occurred, 5 or 6 years ago that most of that was done. So there were some serious questions raised about, is this a responsible action for a company whose interest is in supporting stockholders to continue to invest in CNS? But I think now we're down to a core group of committed companies that are continuing in CNS, and I think a single win is going to make a huge difference for us. So looking forward to the moment that we can announce that. In terms of the sort of the artificiality of saying, this is an academic-based compound and this is a biotech compound, you're absolutely right. This is an ecosystem. right? And I can't think of a single molecule that started in a lab and wound up with the same group of people at the end of the development period. So this is an ecosystem where the compounds are discovered and then they're handed off. Maybe there's a spin-off from the academics, and then there's a bigger biotech, and then there's a pharmaceutical company. One of our problems, I think, is could it be better organized? Right now it's kind of hit or miss as to whether or not these compounds are systematically passed along with the greatest efficiency that we could achieve. And I don't think that's happening right now. So I think your point about this ecosystem is so important, because we need to think about how to have that happen more systematically and faster.
Jeff, one last question from me before we move on. I was tempted to ask you what your top priority targets are, but I won't do that. I think I'll phrase it a different way. Do you think that there are potentially promising aspects of biology that we should be pursuing for their therapeutic implications that are kind of under-resourced now? Or underinvested in?
Of course, the easy answer, Richard, is they're all under-resourced.
Yeah, well, anything in particular. I mean, I'll give you my views. My favorite is ApoE, but
I think we're all thinking a lot about tau right now, and I'm excited about the A beta compounds. By the way, I think we're really seeing biological activity there that's likely to be clinically meaningful, and so I'm excited about that. But I think looking at tau, because of its close relationship with cognitive decline, is a really important direction. And Meiyu is going to tell us about the microbiome. We're seeing microbiome studies across ALS, Parkinson's disease, multiple sclerosis. I think the microbiome is also starting to be a very interesting area of focus right now. So those stand out to me, but there are many other worthy processes and targets as well.
Well, that's a great segue to the next presentation from Meiyu Geng. And Meiyu, we're eager to hear about your program.
Yes. Okay. So I just want to share with you about the 22 years of journey of development of Geneva-1, a marine-derived oligosaccharide anti-AD drug, which was launched last year, as I just mentioned. And the IND filing for the global pivotal phase 3 trials, named as Green Memory, was cleared by FDA on April 3rd of this year. The target population is still on mild to moderate AD patients, and the treatment duration is a 12-month double-blind period, followed by a 6 months open-label extension, which will enroll about 2,000 patients, covering about 200 clinical centers worldwide. And I just want to share with you about the first patient. The first screening has been finished on the October 27th, and yes. And it's the first pivotal study, including the dysbiosis of gut microbiome and the peripheral inflammations, and also the metabolic disorders as the exploratory endpoint. And using the Geneva-1 as a probe, we found that the Alzheimer's disease is not only the local neurodegenerative lesion in the brain, but also is an aging-related systematic inflammatory disease associated with the dysbiosis of gut microbiome and metabolic disorders and systematic inflammations. Importantly, we find that the Geneva-1 can reconstitute the gut dysbiosis of the gut microbiome and inhibit the infiltration of peripheral immune cells into the brain and decrease the neural inflammations. And meanwhile, the Geneva-1 can inhibit the A beta plaque formations and tau hyperphosphorylations in the animal models. And all this together account for its improvement of the cognition impairment. Given the AD is aging-related systematic metabolic inflammatory disease, we thus hypothesize that the working model for the AD pathogenesis, that is, the aging-related injury and the subsequent chronic low-grade inflammation, could lead to the constantly fight between the injury and repair, pro-inflammation and anti-inflammation, which might lead to the high energy consumption of the human bodies. And this would require the gut microbiome to produce more metabolites for energy expenditures to either specific subtypes of the immune cells or injured epithelial cells or epithelial cells for the immune responses, and even for the proliferation of the damaged tissue cells. Based on this understanding, we think that the treatment of chronic complex disease like AD requires a holistic-based strategy rather than the target-centric strategy. A great focus on peripheral multi-organ interactions driven by the dysbiosis of gut microbiome, especially the interaction of the immune and the metabolic systems, will largely advance on our understanding of the pathogenesis and thus open a new avenue for AD treatment. Thank you.
Thank you. That's a terrific presentation. A lot of questions come to mind, but and I appreciate the fact that you've linked Alzheimer's disease back to its primary risk factor, which is aging. It is a disease of aging, and I think many of us would agree that to understand Alzheimer's disease, it would be helpful to have a better understanding of those things that happen as a part of the aging process. But you've opened up this vista that is really looking at aging kind of holistically. And many of us in the drug development area like to think that we gauge the potency and so forth of the molecules that we test by looking at biomarkers. When you open up your targets to things that are sort of aging related, how do you think about biomarkers as a way to gauge the potency of your approach to Alzheimer's disease, since you're looking not only at the brain, but sort of holistically at the body?
Yeah, actually, it's really a good question, given if we think that the Alzheimer's disease is really age-related disease. So in this way, we should have to think about what the origin of the origin of the onset of the Alzheimer's disease. And based on our understandings, we have already finished about 500. The cohort studies using peripheral, the blood peripheral features together with that of the gut microbiome. We can clearly identify the NC from that of AMCI and from that of mild to moderate AD patients. And this is really quite clear that, this can tell us the causal link or causal effect, causal link between the dysbiosis of a gut microbiome and pro-inflammations, and with that of the pathogenesis of Alzheimer's disease. So based on this understanding, I think the peripheral features, including those of the multi-layer and multi-scaled multi-omics approach for identifying the specific immune patterns, with that of the specific, the involvement of the dysbiosis of microbiota. And we can use the drugs like the oligosaccharide and even the natural products. They can from the holistic view, to reconstitute not even one species or one genus of the gut microbiome. It can really make the reconstitutions of many of the species of the gut microbiome, and in that way, function as a whole, it can really decrease a lot of inhibitions of the metabolites that generated by the dysbiosis of gut microbiome, and then that can decrease the inflammation, which can driven by that of the disordered metabolites. So that is, I think this is a holistic view.
Thank you. Any other questions from any of the other panelists for Meiyu at this point?
I have one question, Richard.
Sure.
So Meiyu, one of the things that we've thought about, and you and I have discussed this actually, but I think it'd be worth hearing your current thoughts. Are the microbiomes across the globe sufficiently similar that you think GV971 will have a similar effect in China, where the drug is approved and you've shown the benefit? Is it going to affect populations in North America and Europe and South America and Africa similarly? Or do you think there's a certain microbiome that's geographically related that might affect the activity of GV971?
Yes, Jeff, it's really a good question, and also it's really a big challenge for us to think about this question now. We know that the microbiome is really have the very diversity, largely across that of the geographic regions, and also the different cultures and different diets. That's just really true. But we learned that it's not only one, the species of the microbiome can only generate one type of the metabolites. There are so many, the species of the microbiome can generate one kind of the metabolites. It's one thing. And another, there's the one kind of the species of a metabolite. The microbiome can generate a lot of metabolites. So in this way, we not use how to identify how many types of the gut microbiome involved in the pathogenesis of the Alzheimer's disease, but to only care about what the final outcome. For example, what the metabolic disorder, what the imbalance of the specific immune subtypes involved in that of the AD pathogenesis. So in this way, I don't think there's a great difference between the Chinese people, Eastern people, with that of the Western people, provided if the gut microbiome, dysbiosis of gut microbiome, is really play very critical role in the AD pathogenesis.
Thank you. That's very informative. We're now going to move on to Dr. Gene Kinney, who, along with his colleagues, has done a remarkable job in establishing a totally new company devoted to pursuing potential treatments for neurodegenerative diseases. And I'm happy to hear from you about your journey with Prothena.
That's great. Thank you, Richard. So just building on some excellent discussions thus far, I wanted to bring this maybe back up to a little bit of a higher level and talk about what we see as some of the opportunity in the area of Alzheimer's disease treatments. And I think we're really at a wonderful time if we look at the pipeline, for example, that Jeff showed us earlier, in a movement from thinking about symptomatic treatments for the disease to a place now where, as Jeff just told us, 80% of the drugs in the Alzheimer's disease pipeline are focused on modification of the disease. And to me, that's a very exciting shift. It suggests that we're moving beyond providing relief of symptoms without regard for the underlying disease pathology and the progression of that, but now focusing on that disease progression to slow the disease process, thereby working better with symptomatic treatments. And we can start to envision where we go with this as well. Richard, I think you asked an excellent question, which was, what are some promising targets as we think about moving forward with therapeutics? We just heard a very interesting one in GV971. But I think we can talk about this in other ways as well, which isn't necessarily about the molecular target, but where the field is going. Because as we think about disease-modifying approaches, and we start to put that together with some of the excellent work that's being done looking at longitudinal progression of individuals that are at risk for Alzheimer's disease, different initiatives like the A3, the A4 initiative, Diane leads, and others, we can actually start thinking about intervening earlier stage and start talking about prevention trials. So secondary prevention trials, primary prevention trials. That's very, very exciting. And ultimately, with the advent of newer technologies and modalities, things like cell therapy and progress that's being made in that space, we can start to talk about regenerative approaches in the future. which could potentially bring patients back to a pre-diagnostic state from a functional perspective. And so, again, very, very exciting. But to do this and to move our pipeline of 121 drugs to something more akin to what we are expecting from an oncology pipeline, like 3,000 drugs in the works, we're going to need capital, and we're going to need to fund that, and it needs to be funded on an ongoing basis. And if we can move forward in the slide here, I want to show you a little bit that we've been discordant in thinking about our funding. And so what I'm showing here in the lower left are two figures that look at private investment. So these would be investments in areas of venture capital, initial public offerings, and even follow-on offerings for publicly listed biotechnology companies. And I think what you can see in the lower left-hand pie chart here is that almost double the amount of funding that occurred in neurology across the board, about $11 billion in 2019. Double that, $22 billion, occurred in oncology. Yet if we look at prevalence and the potential impact, you can see the disconnect here. So the largest indication in oncology around the US is lung cancer. And you can see the prevalence in 2019 over the same time period was about 541,000 individuals in the US. By contrast, Alzheimer's disease over that time period, about 5.8 million. So why might that be, and what might be happening? And I had an opportunity ahead of this discussion to reach out to some investors in the space. So one investor in particular, manages a healthcare-focused investment company that has under management somewhere between 1.5 to 2.0 billion dollars under management. And he really pointed to three things. The first, subsets and segmentation, which I think was an interesting concept. And to quote him, he views investing in Alzheimer's similar to investing in oncology 20 years ago. And just like we know now in areas like lung cancer, that it isn't just one cancer, but in fact driven by different mutations, it's likely that we'll learn the same and start to think about Alzheimer's disease in a similar way. And I think this really brings out the idea of the need for patient selection and the use of biomarkers in doing that. A second topic he really focused on was this idea of a back-end loaded risk profile for Alzheimer's disease. And I think this goes to an earlier conversation we were hearing in the panel between Rachelle and Jeff. Where does the funding come from? And I think you do see transition of risk as these new molecules are being developed and brought through the development space from areas like the academic labs to the biotechnology companies to large pharma. And in part, that reflects long trials in disease-modifying therapies for Alzheimer's disease, expensive and very robust investments that are required to get to an answer about whether the approach is going to be successful or not. And the back-end loaded nature of the return on that investment is certainly something that the investment community sees as a potential issue for the initial investments early in the scientific journey. Specifically, things that were brought out in that discussion that I had was the lack of predictive animal models in the Alzheimer's disease space, the lack of FDA guidance and therefore a high regulatory risk that's retained with any investment, and the lack of surrogate efficacy biomarkers. So three things specifically brought up. And then the final point that I just make on this is I think Jeff made the point that he remains optimistic in A-beta targeting approaches. I share his optimism in that space. And I think, the approval of a first disease-modifying agent, whether that be A-beta targeting or not, I think will be very important from an investment consideration. And just to quote the investor that I spoke with, quote unquote, I think that FDA approving aducanumab would be highly beneficial for investing in the disease, similar to how Sarepta approval in DMD opened the floodgates for investment in muscular dystrophy research. So if we move to the next slide, what can we do? And I think, Rachelle will talk about this in her segment of the discussion, but I think improving trial success and moving the risk profile earlier into the development path so that we understand the likelihood of success earlier is a critical component of what we need to do. Obviously, deploying biomarkers in terms of selecting the right patients, those patients that are most likely to respond to the therapies that we're testing in any given clinical trial, selecting the right endpoints that match that patient population. It does no good to have a clinical assessment scale that's specific for very advanced patients put against a very early population, for example. Having the right molecule to test the biological hypothesis, and of course, testing the appropriate biological hypothesis. I think that one thing that we do need to be careful of as a field is ensuring that when we do have unsuccessful trials, that we're careful about dissecting the reason for the lack of success in those trials. It's all too easy for individuals, particularly in areas that aren't steeped in science every day, like an investment community, to default to the idea that a failed trial is because the biological hypothesis was wrong. But in fact, it may be because we chose the wrong patient population with which to test that hypothesis. And I think we need to be very specific about how we talk about these. So as we iteratively move these programs forward, learn from prior trials, and employ those learnings moving forward, we can communicate that efficiently and keep the investment flowing through. So with that said, I'll pause here and look forward to some dialogue around this.
Thank you, Gene. I'll defer to the rest of the panel if you have a question for Gene, and then we'll get to Rachelle. I think he's raised a lot of interesting things about how do we finance this whole operation, because it is expensive.
Gene, I have one question. How do we manage the expectation of venture capital and investors for relatively short turnaround time when we have, an 18-month trial and a 24-month recruitment period? We're almost out of the window for the expected return just in the length of the trial. So are there longer-term funds? What can we do about this contradiction?
Yeah, I think it's a great question. And I think it is about bringing the expectations into line. So I think one can, start to even incorporate either biomarker-based approaches or even clinical-based approaches earlier in a clinical program. But the expectation, for the reasons you just laid out, can't be that, only an alpha value two-sided of 0.05 at 90% powered matters, right? It has to be that we are directionally learning how to power our phase 3 studies from an effect size perspective. And I think there has to be preconditioning done that's what the trial is designed to do. That's what we're looking for. The marker of success is de-risking your larger phase 3 18-month trial. And we have to move away from a place where we say, knowing that we've influenced the biology by some marker of target engagement is sufficient to move forward. But at the same time, that the expectation isn't that only a P of 0.05, robust effect size is enough to move forward and fully test the hypothesis in larger trials over larger longitudinal periods.
Great points. I'm sorry. We're going to give Rachelle a chance to get her presentation and maybe have a little chance for discussion later. I'll turn it over to Rachelle.
Thank you, Richard, and thank you to all of the prior speakers. What I'd like to talk about is innovation. That's the title of our symposium, Scaling, Linking, and Creating Action to Drive Global Innovation in Alzheimer's Disease. And I spent a little bit of time thinking about, what would really bring this all together and be innovative? So what I'd like to talk about is the architecture that we need to create in order for all of these many activities and advances that are taking place to come together for patients in a timely manner. We need integrated solutions, and those integrated solutions should aim to bring better treatments to patients at a lower cost to society. If we don't bring better treatments, then nothing that we do matters. if we don't lower the cost to society, people won't have access to those things that we create. So in this architecture, I think the components that are needed, you can have many pillars in architecture that are not silos. Okay, so they have to interact with each other, but they can still exist as pillars which hold the structure together. So we have pharmaceuticals, and we've talked a lot about that, both Jeff and Jean, and then in a very specific way, Meiyu, have talked to us about molecules. What we didn't talk about yet is delivering those molecules. So one of the many challenges that we have as drug developers in Alzheimer's disease is getting our molecules to the brain. And so as we're thinking about the molecule in its very earliest stages, we also have to be thinking about the delivery system. And it's almost a parallel drug development process that goes on to try to enhance delivery of, say, large molecules to the brain, or small molecules that by their nature and structure might not enter the brain easily. And we also have to think about the ecosystem. And in the case of development of drugs, that's the ecosystem of clinical trials and clinical trials designs. So every design decision that we make affects that timeline that Jeff was talking about, of how long it's going to take to recruit the subjects, how long it's going to take for people to be in a double-blind condition in a clinical trial. And then even, what do we do after they finish the trial? Increasingly, there's the need to learn more. Okay, so just a little bit about each of these points. Most of the disease-modifying approaches at least started out as large molecule antibody approaches. Jeff showed on his slide that there are now many small molecules as well. We need to develop things like brain shuttles that bring molecules into the brain. We've shown already that you could increase the amount of antibody that gets into the brain by up to 15, 20-fold with a brain shuttle approach. How do we design our systems, though, so that we can be studying the molecule attached to a brain shuttle at the same time as we're studying it for safety and efficacy? We can't do these things in a linear way, or it will take us all of our lifetime to develop just a few things. And so in terms of trials designs, increasingly what we need to go to is platform type trials. I think that we could conceive of trials right now that are both basket trials and umbrella trials. Basket trials being that we are trying the same drug in multiple different populations. So Jean talked about the importance of matching the population to the trial. What if we had trials that somehow were running simultaneously in preclinical AD, prodromal AD, and mild AD? That's doable, but we haven't done it yet. So in the future of integrated solutions, the development of pharmaceuticals needs to change. At the same time, there's the whole world of diagnostics. And fortunately, I'm in a place where we work on diagnostic tests as well as pharmaceuticals. But how do we put these two together as pillars that lead us to integrated solutions? Well, one was also mentioned, and that is blood-based biomarkers. We can develop ways to identify subpopulations, either for clinical trials or for ultimate matching of the patient to the therapy. But as we develop these in thousands of laboratories all across the world, we have to develop them in ways that are replicable and that are doable everywhere. And so automated approaches to biomarker measurement are increasingly important because, you may want to develop a biomarker for a particular drug development program, and you may be able to do that successfully in-house or through a vendor or in an academic laboratory. But if other laboratories cannot get the same result using the same procedures, this test will have a very limited utility for developing your therapeutic. Also, we have the possibility of increasing the precision of what we do and increasing the number of observations that we can make with respect to outcomes through digital approaches. This field is exploding. Fortunately, many players of all different types, from giant tech companies to individuals, are working on digital outcomes of different types. But we have a tremendous lack of coordination and a tremendous lack of guidance from our regulatory authorities about how to go about this. They are trying. They are overwhelmed with the number of things coming their way. But even questions about, say, version control. When you have something digital, you're always changing it and upgrading it. And how do you deal with tools that change, or tools that you change on purpose through the next pillar that I'm going to talk about, which is insights. Okay, so we have pharma and we have dia, and we have to put them together to get to integrated solutions. What do I mean by insights? Well, in the process of doing our work, which takes decades, right? Developing drugs takes decades. Once we have the drugs, if they're helpful, they're also used for decades. So a lot of information is generated, the kind of information for which you can use advanced analytics or machine learning or other ways of looking at condition and response. You're giving a drug and you're getting a response over time. If we were better at curating individual patient data, in other words, all the data pertinent to a patient, whether it came from their smartwatch, whether it came from a screening tool they took online, whether it came from a physician's office or a different physician in a different state where they used to live, if you could put together all the data on every individual patient so that when the patient needs the data or when their current physician needs the data, they are there, and at the same time, generate meaningful data at scale. This is a tremendous aspiration that's completely doable. What about personalization? Okay. In order to save cost, we have to get the right drug to the right patient at the right time. The way we do it now, it's wasteful. A lot of people try a drug that doesn't work, or a lot of people don't get the drug at all because they don't have access to it. Through an integrated development approach with integrated solutions, we build into our early discovery which patients match best with the drug and predict who's going to have the most meaningful outcome. And then we use our meaningful data at scale to test those hypotheses and to refine those algorithms. So in the end, what our goal is to have integrated solutions. Instead of throwing a lot of new innovations out to the world, throwing them out to patients and doctors and saying, do the best you can, we want to integrate the solutions as we develop them. We don't want to leave anybody out. These solutions are for people at risk for Alzheimer's. They're for people with prodromal AD, mild, moderate, and severe disease. If you have Alzheimer's, you're eligible for solutions. And we want better treatments for all of those patients at a lower cost to society.
Thank you, Rachelle. It's very, very informative, very thought-provoking. We have just a couple minutes left, so I'm going to ask, in the spirit of this meeting, whether or not people have specific and hopefully very pithy thoughts about what we can do to issue a call to action to move this field forward more quickly. Maybe we could start with Rachelle. Do you have a specific thing that could be done?
I think, I've said many times that one of the keys is getting rid of the artificial distinctions, okay? The distinctions that we talked about before between academic and industry, biotech and big pharma. It's going to take all of us doing everything that we can do and sharing it with each other to solve this problem. So taking down the silos, whenever we are hitting one of those architectural barriers that we think can't be changed, let's get a consult and see about changing it.
So thanks. Anybody else have some final thoughts about things we could include in the report from this meeting that would be a call to action? I have one thought, just maybe people give a quick reaction. Is it possible that we could call for co-funding between government entities and particularly smaller companies undertaking high-risk ventures that have the potential to benefit society? Is that something that people would think is possible or worthwhile?
I was thinking the same thing, Richard, that the dialogue between the private sector and the major government funders, I think, is not particularly strong at this point. I think those communication channels are not as influential as they should be. I think Rachelle just articulated a wonderful sort of research agenda and how to think about it. And part of that will be executed by federal funding, but I don't see that dialogue going on in a meaningful way.
Any other final thoughts from either Gene or Meiyu?
Well, I would agree with what was just said. I mean, I think the idea of bringing together funding sources so that we can actually distribute risk across the various constituencies, I think is an excellent idea, and I think one that folks could get behind. I mean, at the end of the day, making an impact in this disease space is so important for patients, but the potential upside value from a commercial perspective is there as well, and should be incentive enough to think about risk-sharing based approaches.
Okay, I think we've come to the end of our time. I thank all of the panelists for very informative presentations, and more importantly, I thank all of you for the work that you do to advance the therapeutics for patients with Alzheimer's disease. We are in desperate need of them, both for individual patients and for society. So I thank you very much for all of your efforts.